said, okay, let's just talk about metals and kidney disease. Let's try to understand what these connections mean. And then hopefully with some of the small studies that are emerging, some of the research that is emerging for CKDU, and some of the more ambitious work that is also ongoing, hopefully we can have some learning and we can have some advancement of knowledge in a few years from now. So here is the periodic table. Most of the periodic table, you can see a large part of the periodic tables are trace elements. Yes?
Sorry, just to interrupt, it looks like your screen may not be advancing right now.
Oh, really?
Yeah, I'm just seeing.
Okay.
Yeah.
So let me just stop. I think I know maybe why. So let me just start again, okay? Sorry for this.
Let's see if that would work.
Let me, hopefully I can, I think it's because I had changed one small thing and that made it share screen. Okay, here. Very good. So let's see. Oops, 1 second. Did it change now?
Yeah, that looks better. Thank you, Anna.
Perfect. Thank you for letting me know. So, okay, many elements in the periodic tables are metals or metalloids, like semi-metals. And I put in red here the metals for which we have a lot of evidence or substantial evidence that they might be nephrotoxic based on epidemiological research and experimental research, especially for metals such as lead, cadmium, mercury, uranium. Actually is quite well known that these are nephrotoxic, at least at moderate to high levels. And arsenic, there is some emerging evidence supporting the nephrotoxicity of arsenic. For tungsten, there is some kind of interesting research done actually by Kathy James here, but because we know so little in general about the health effects of tungsten, I think this is really not well known yet, what the role is of tungsten in kidney disease. So, but I want to focus on some of the metals for which we know the most. So lead and cadmium are very interesting, as both of them are divalent cations with very long half-lives, very important from a human exposure side, as you know, from leaded gasoline, lead paint in the US, and many other sources, which are legacy sources as well as important sources remaining today. I think something that people don't understand is that, or seem to ignore or be less aware, is that lead production continues to grow in the world. Lead continues to be used in many different products, batteries, and other things, and lead is still to be mined and produced globally. And cadmium, you can see in the figure, lead production has become stable even during the 21st century, and it's still very widely used in many numerous products, including fertilizers, which might be particularly important in agricultural communities. And this is because the phosphate rocks that are used for fertilizers are naturally rich in cadmium. So these are very important metals that have very, very long half-life. So basically, once we absorb them into the body, we practically cannot eliminate these metals. They are going to stay. Lead behaves like calcium, is stored in the bone. Cadmium behaves a lot like zinc and is going to be stored in soft tissues, the kidney in particular, and the liver and many other tissues. So these are really, really important metals. And for arsenic, which is the other one that I mentioned, we have quite emerging evidence. It's particularly important in groundwater. And you can see that in the western part of the US, arsenic is very commonly found in groundwater. Communities relying on groundwater for drinking water are naturally exposed to quite high levels of arsenic. And if you continue going through this map, which is kind of artificially cut here at the Mexico border between the US and Mexico, this obviously continues going through Mexico and Central America. So arsenic exposure is just naturally common in groundwater across Central America. In addition, rice is another important source. And arsenic is very different from these divalent cations that I mentioned earlier, cadmium and lead, because arsenic is not accumulated in the body, but arsenic is excreted relatively rapidly. In just a few days, you are going to excrete it. And this is because even though inorganic arsenic has a relatively slower excretion, inorganic arsenic is going to be methylated, and the dimethylated forms of arsenic are going to be excreted throughout the urine. But if you drink contaminated water every day, then you have a persistent exposure throughout decades and a long time. So this is still a very relevant exposure. So this is a figure that, as you can see, it says here is the courtesy of Dr. Virginia Weaver, and where it shows where different toxicants and where do they affect the kidney. And you can see that metals are particularly harmful for the proximal tubule. And I think this is very relevant for CKD-U, even if we don't really know how much of a role these metals play in CKD-U, but it would not be a surprise if they play some contribution, given that they are really toxic at that part of the proximal tubule where it seems that CKD-U is starting. It might be that this is the first place where the harm develops. So, okay, let me go back. And so in this slide, it's showing more about lead nephropathy. And as we mentioned, lead is affected from the, it affects the proximal tubule. This was kind of discovered. Of course, there are many, many examples of how lead was related, and many epidemics, many outbreaks have been described in occupational cohorts, in other studies. But one of them, which is very striking, and I think it might be relevant for CKD-U too, is this outbreak in Australia related to lead paint. And the reason why is because these were children, these were young people, and you could see a clear epidemic going on in Queensland, in this region where they had the lead paint. And even children in the third to fifth decade of life, relatively young people, young adults who had been exposed as children had CKD very early. And I think, again, if these exposures are happening during early childhood, during youth, during childhood labor, combined with other factors going on, it would not be a surprise that this disease kind of starts prematurely. And so this is a slide also put together from an up-to-date review that Dr. Weaver prepared together with Bernard Jarr, who is also a nephrologist from Hopkins, and describing how lead nephropathy looks like. And you can see that this early phase, there is some pathology that can already be identified with these inclusion bodies very clearly in the proximal tubule cells. And you can start seeing these increased levels of low molecular weight proteins, amino acid, glucose. And so this is what happens early on, which might have some similarities with CKD-U. And then you advance into the more chronic phase where you develop the chronic renal failure with tubulointerstitial fibrosis. And so this is interesting. We don't know if this is relevant for CKD-U, and this is some of the mechanisms for lead toxicity, which is very related to oxidative stress, and because the mitochondria are very important, and the proximal tubule is a place very active in mitochondria because the mitochondria are very highly needed for the active transport that is going on in the proximal tubule. So this really makes a part of the body that is particularly susceptible to the toxicity of lead. We know lead is toxic for many organs and systems, and of course we have mitochondria everywhere, But you can see if that's involved and oxidative stress is involved, you can see that the places where there is a high accumulation of mitochondria might be some of the areas that really you can see the disease earlier. So the binding of lead to the replacement of calcium by lead is very well known, and this is why lead accumulates in the bone. But for instance, it has been very well known that lead binds to calmodulin, which is an enzyme, a protein that really needs calcium for its function. And it has been shown in some animal experiments that this seems to be one of the mechanisms for the very well-known neurotoxicity effects of lead, because calmodulin is very important also for neuronal transmission. And so this could be, and this also has been involved, this mechanism has been involved also in potentially in left ventricular hypertrophy and heart failure types of effects of lead, because calmodulin also has been very well known to be involved in heart function and ventricle function. So, and this could be another mechanism also relevant for the kidney pathways, for the kidney toxicity. The other one mechanism that has also been proposed that could be related to lead-induced nephrotoxicity, that could be actually also relevant for CKD-U, is related to the lysosomal membrane permeabilization with lead affecting lysosomal function. And so these are kind of interesting mechanisms and papers out there that have reviewed this quite carefully, that I believe also could kind of be interesting to study in the context of CKD-U. Okay, so this was lead, which is a very well-known toxicant. And then I want to talk a little bit about cadmium, which has some similarities with lead in the sense that both of them are divalent cations, both of them are toxic, have no essential function, but interact, replace our essential metals. Both of them, something very interesting to understand for both cadmium and lead, is that life on Earth, the biosphere in the planet, didn't evolve with exposure to these metals, because these metals were really deep in the ground. And it's only from mining that humans have really contaminated the surface of the Earth, which has been really important for humans, but also for ecosystems. And we know that these metals have had major implications for all kinds of other animals and plants. And so, but cadmium, one of the first times that we learned that cadmium was nephrotoxic, because cadmium was only discovered in the 19th century. It was discovered because it was an impurity of zinc. When they were trying to mine zinc, cadmium would show up and they would realize it was something similar but different. And then cadmium was, because of all the properties that I described earlier, like great for batteries, great for as a plasticizer, a great pigment that gave beautiful colors. For all of those reasons, it started to be produced and mined very extensively, especially in Asian countries. And today in the world, actually cadmium toxicity is particularly important in Asian regions, in China, Japan. And in Japan is where they found for the first time that cadmium could be very toxic to humans, with a very painful disease, which was due to a bone effect, and also characterized also by proximal tubule damage and declining creatinine clearance. But end-stage renal disease was not so common. And I think that's kind of interesting also for us to think about potentially. And another study done in Thailand, in this study in Thailand, in the Mae Sot district, we can see it was quite a number of big sample size. It's very prevalent, the high cadmium, a lot of high cadmium toxicity, like people are really exposed to cadmium because of the zinc mining and rice contamination. And you can see that the people over time, in just 5 years of time, these people who have been exposed to cadmium are increasing the beta-2 microglobulin levels, which has been related to cadmium with very strong evidence, the total protein, the serum creatinine is increasing rapidly, and glomerular filtration rate, the prevalence of eGFR below 60 is increasing very rapidly, something that would not be normal in just 5 years for a population. So again, showing that cadmium is nephrotoxicant. And but the early detection might be very difficult, and really these metals need to be much better studied. And even though we have some good knowledge of how cadmium could be related to these proteins, that might be related to the tubular effects of the metals that could also be relevant, and the acute kidney injury markers that have also been shown to be relevant for CKD-U. So I want to show a little bit of a study that we did, actually, myself with Dr. Weaver. We published, this is an old paper in NHANES, but we found clear associations with cadmium. We found maybe not so clear association with lead, some trend. And also, this was with albuminuria. This is an association, very clear association with albuminuria, and maybe less clear, but some trend also, significant trends for eGFR. But this is cross-sectional, so this is very limited in a way, and we really need prospective studies. But when you do this joint analysis, it seems that both the high blood lead, people who had both high lead and high cadmium had more risk of chronic kidney disease, and this was not a significant interaction. It was just additive effects. This was just the two metals being additive. So if you are exposed to both, your risk is much higher because you are adding the risk. It's not synergistic in the sense that it's not a multiplicative risk, but it's an additive risk. So this is also something important for CKD-U to think that all these risks might be cumulative. You might have heat stress, pesticides, metals, nutritional issues going on in early childhood, and many other things going on. And these are independent effects that are adding up. So your risk is just kind of going over the threshold, and you are much more likely to develop these diseases much earlier in life. And I think you don't really need multiplicative interactions to have an excess risk that is quite substantial when you have these additive effects. So this other work that we also did with Jeff Fadrowski, and who is a pediatric nephrologist at Johns Hopkins, and with this study, the interest here was to look early in life, because very often people say, okay, you are studying these metals, but people have kidney disease, people have diabetes, people have all these other risk factors. You cannot really disentangle what is the effect is. And so for this reason, we tried to do this work, and Jeff was the leader of this study, looking at what happens when you don't have diabetes, you don't have hypertension. Can we see an association with these metals very early on? And exactly that's what we found. And both with cystatin C-based definitions and creatine-based definitions for eGFR, and we could see that there was a decline, and there are multiple models with different levels of adjustment, but the association was quite robust with a decline in eGFR with increasing levels of lead. And so this was quite interesting, and especially with the cystatin C-based equation, which is interesting. We don't know if this is truly related to function, renal function, or is it something else related to the signal of cystatin and what does cystatin mean versus creatinine base at the same time, it could be more measurement error because of reflecting other things that are also not kidney function. So this is, the jury is still out of why we see these differences. Yes, is there a problem? Oh, sorry, I heard somebody talk. Very good, wonderful. Okay, good, so good, this is just basically what I said. Very good, so this is kind of my high level picture of things and things that we've done, and this is not updated research in the sense that there are other studies also more recent and in other populations done that confirm really that these metals are relevant, although they are not prospective. I forgot to add some recent studies also showing that lead in the water, one of the big questions is that what if these biomarkers are affected by kidney disease, and all this association that we see are spurious because of this kind of reverse causation. And there was this paper published recently in one of the kidney journals showing that even lead in the water in the US, which cannot be affected by kidney function, is related to more severe kidney disease across the nation. And so that's quite compelling evidence, again, that even at these low levels in general populations exposed to these metals matter to the kidney. But I want to talk a little bit about arsenic. And so as I mentioned, arsenic has a relatively shorter half-life. In the Stronghart study, that is a population-based cohort across the Northern Plains, Southern Plains, and Southwest tribal communities, people had very constant levels of arsenic in the urine. This is levels in the urine over a 10-year period in the three communities. And you can see that all the variability is across people. You have very little within people variability. And the reason is because, again, people are drinking the same water source, which is the major contributor. And so this is very, very chronic. We are capturing a 10-year window, but probably these people have been exposed to these levels for many decades. And we are learning that this has very important health effects in many systems. And this work is possible because of a participatory approach. This is a picture from a couple of years ago, just before the pandemic. We meet every year, once a year, and we haven't been able to meet in person. We are planning now to meet again. But the local contributions, the local input from the communities, the local feedback, how do we report back, the data sovereignty, because these are tribal nations and the data, it's on loan to the investigators. So these are really, really important issues that I believe there is also a lot of connections and similarities with the communities that are affected by CKDU. So this is the design of the Stronghart study. It's a very longitudinal cohort going on for many years. And I think one of the lessons is that we need to be very patient with these cohorts. There is so many players, but these are very, very important efforts to keep the cohorts going. And I write here that funding is critical to maintain a project and research. And you can see in this cohort that at the beginning, it was very well, like every few years, very well planned. But then in the second phase, when the family members were recruited into the cohort, suddenly you have gaps of almost here a gap of 10 years. And now actually between phase 7, we are going to start phase 7 now. So you can see very big gaps in research. And the reason is because of changes in priority of funding. And we need to be aware about that as scientists. And these are going to be limitations in our research. But this is the overall framework that we use for our prospective cohorts. We use the metal biomarkers, which are great because they integrate multiple sources of exposure from water, air, food, soil. We are able to connect this information with clinical disease, which is our ultimate goal. But also we have this mechanistic information that we can gather, especially now with all the omics technology, this is really a great opportunities to look at mechanistic research and pathways that are relevant. And then, of course, we need to always think about these co-exposures, these even additive effects that could have with our exposures of interest, in this case, the metals. So this is our overall framework. And we've used this framework to study kidney disease in several of the studies in the SOHOR study going on. For instance, we looked at prevalent albuminuria, where we found an association. We also found an association with incident albuminuria, although the sample size was small, and it ended up that we never published the paper, which sometimes happens. And then this other paper where we look at chronic kidney disease, looking both at prevalent and incident. And I think this study is particularly interesting and relevant because, again, when we have a cohort study, what happens at baseline might be very different than what you can see when you start looking longitudinally. And I think we need to be very aware of these cross-sectional biases, especially reverse causation that I've talked earlier. So in this figure is the cross-sectional and prospective analysis for arsenic and reduced EGFR with the, I think it was at the -60 definition cutoff. And the association is for the cross-sectional analysis on the left side and for the prospective analysis, for the prospective cross-sectional analysis in the top and the prospective analysis at the bottom. And you can see that when we look, the sum of inorganic and methylated arsenic species is the best biomarker that we have of exposure to inorganic arsenic, because as I mentioned earlier, inorganic arsenic is methylated in the body. So you need to look at both the original inorganic and its methylated metabolites. So when you look at that, we could see this clear kind of reverse causation looking association. So people with having higher arsenic were less likely in the urine, were less likely to have kidney disease. And the people with more likely to have kidney disease had the lowest arsenic level. So people who cannot excrete arsenic, because the kidney is the primary organ for arsenic elimination. So we are not able to eliminate it. And this is especially true. This inverse association was actually mostly related to inorganic arsenic. So it's actually flat. There was no problem for the methylated species. The methylated species can be filtered through the kidney, even if the kidney is not working well. But if your kidney is not working well, your inorganic arsenic is not going through the kidney. And you have this very remarkable inverse association, which is most likely reverse causation. But that's what's happening at baseline. Now you look what's happening prospectively, and you even adjust for baseline EGFR to make sure everybody starts with the same EGFR level. And now you look at the prospective effect. And what you see is a completely different picture. So even if when you were not able to excrete if you had a problem at baseline, once you remove that, arsenic actually looks quite toxic. And actually DMA was in this case what looked the most toxic. Although, DMA is the primary metabolite of inorganic arsenic. So I would just say it's in general inorganic arsenic exposure related to increased risk of kidney disease. Okay, so I think I need to start wrapping up. Sorry, I was a bit slow. I wanted to say that genetics is very important to study. For arsenic, is particularly relevant because there is an enzyme, methyltransferase, that is involved in arsenic elimination. In arsenic methylation, sorry. And we have very, very strong genetic signals that this is important. And we have evidence that there might be an interaction between this gene and arsenic methylation for HOMA-IR. Of course, this has not been replicated in another cohort. We need to be very careful. But again, and this is not a kidney outcome, we would need to look at kidney outcomes. But again, this shows that this is important. And here I have an example, a paper from another colleague, a very close colleague, Dr. Telles Plaza and her team. This is a cohort, a general population from Spain. And in this study, the interesting thing is this genome-wide analysis, or close to genome-wide, and some clear genes were strongly associated with urinary cadmium. And so this was very interesting to find out, or I think this was related, sorry, to albuminuria. But when we look at the interaction, what is interesting here is that there was a triple interaction between cadmium, one of the transporter, which is a transporter involved in the endosomal zinc, so in zinc transport, and then another gene involved in the transport of tubular albumin and in oxidative stress, endothelial damage and dysfunction. And if you have this, these different SNPs combination, which was only 14 people in the study, but here we have a multiplicative risk. Here people are much more likely to have albuminuria. So there is a very clear dose response and a very clear multiplicative risk. So again, this showed us that there is so much work that needs to be done to look at gene environment interactions. But again, these are tiny studies. We need big data, we need big consortia to be able to do this work really well. So a lot of effort is needed, and Dr. Telles Plaza, she's very interested in this idea of these metal GWAS and try to pull cohorts together to advance this work. Okay, I want to talk a little bit about CKDU and what do we know about CKDU and metals. And there is some work out there, which I think is quite interesting. And one of the studies, it's that I know Dr. James is involved, and other people that might be here in the call, was done in Guatemala. And it recruited the participants during the sugarcane harvest and then off season. And they were really studied multiple times. You can see the sample sizes get small over time, and they measured several metals in the urine. And what is interesting in the study is that they found relatively robust associations. They run like a number of sensitivity analyses, and urinary arsenic and urinary cadmium were related to lower EGFR, higher NGAL, which is again this marker of tubular protein, of tubular interstitial disease. The albumin signal was not very clear. And for nickel, it was not clear either. So this is kind of interesting findings, again, relatively small sample sizes. And another cohort also has found nickel. In this study, nickel was measured in the urine. Here they measure nickel in toenails, and they measure other metals in toenails, but metals in toenails are very tricky. For instance, lead doesn't really get incorporated into the toenail, so measuring lead in toenails is generally not very helpful. But you can see here there was quite a clear association between nickel and serum creatinine, but you can see the sample size is tiny, 39 participants. We need larger data to really be able to do this work well. So in the last kind of 5 minutes or so, I want to talk about how to assess metal exposure, and thinking carefully what are the advantages and what are some of the issues. And as I mentioned, the good thing is that we have really good individual level measures. They integrate sources of exposure, and they can really reflect long-term for some metals or ongoing, depending on how the half-life and how the exposure is. And for arsenic, we have this marker of biotransformation. But we need to be very, very, very careful in the interpretation of metal studies, especially nowadays with these metalomics opportunities, the big assessment of large panels of metals. There is a real danger of being very quick and not thinking carefully of what these levels really mean. How much is it exposure, but to how much is it how the body is handling? And so we need to be really careful. So for arsenic, cadmium, and lead, this is a table with some of the specimens. Where can we measure it? Generally, arsenic, it's in the urine, in blood, is very low levels, not very helpful. Cadmium is generally very well measured in the urine. You can measure it in blood also. It brings you a different information. And you can see that the cadmium in the urine has a half-life of decades. For lead, bone can be a very interesting biomarker through a K-shell X-ray fluorescence. But I would think that for most cohorts, especially, it's very complicated to do with a lot of measurement error. So I don't really think this is ready for low chronic exposure. Is difficult to do. But in blood is very well established. Something interesting about cadmium, and sometimes people are really surprised that you can measure cadmium in urine and that it has this very long half-life. So I wanted to show here this cartoon that shows what's happening when cadmium gets absorbed into the body, generally through the gastrointestinal tract, and very often it goes through transporters like for iron. It's going to induce the induction of metallothionein. And the reason is because cadmium is so similar to zinc, that metallothionein, which is expressed by zinc, starts to express itself even more. And that has been shown that that's a good thing. It protects from cadmium toxicity. So in a way, that's a good thing. But now this cadmium metallothionein complex travels throughout the blood and gets accumulated in the renal cortex. And it stays there, it's not really excreted. But because it's in equilibrium with the urine, the levels in the urine reflect not recent excretion, but really the long-term concentration in the kidney cortex. So that makes cadmium like an interesting biomarker, but still complicated. And I think we need more research, especially at the low levels. So I want to kind of end, and I wanted to say that when we do this type of research, really, we want to make sure that it's relevant for our participants in the study. These are serious conditions. We need to make sure they get the feedback that they deserve, but also at the community level, at the regional level, and how we can inform countries and more global policies. Oops. So kind of in summary, the evidence supports that these metals are relevant for chronic kidney disease at levels of exposure that are common for general populations, but we need much more research, both for general populations as well as for CKD-U in particular. So this is an area of research that is very active right now, and it's exciting for us to be involved as part of the CURE consortium and other cohorts. For instance, we are working now with the MESA cohort, which is a very new population-based study that has more than 6,000 participants. And we've completed now 90% of the work. Will be done very soon. With the pandemic, we suffered some delays, and that's why I was not able to show you any data. But I can tell you that in the preliminary analysis that we've run in MESA, we can see that these metals matter for kidney disease. So even for general populations here in the US. So this is really to be continued, very active area of research. So I wanted to acknowledge the participants, communities, funding organizations that make research possible. I think I need to add the CURE consortium logo here to acknowledge this new source of funding. And I wanted to also acknowledge some of the nephrologists that I've worked with over the years, with Sumit Mohan at Columbia now, who is the PI of the field study in India, which is part of the CURE consortium, as well as colleagues that I've mentioned throughout the talk, also like Jeff Fedorowski, pediatric nephrologist, and Qingqi Kuo, who is also a nephrologist in Taiwan, and Jason Newmans, who is another nephrologist that we work with as part of the StrongHeart study. And also, as I've acknowledged over the slides, of Dr. Virginia Weaver, who really introduced me to this field of metals and kidney disease. And I think I am done with the presentation, and I hope we have some time for a discussion and comment questions. I would love to hear what people think and what their experience is from others, and many other research opportunities that I've left behind, and I would love to hear more about them.
Thank you so much, Anna, for this great presentation. It was so clear and very informative. And also you showed the evidence about the gaps that we need to continue developing research on that areas for generating more knowledge about the relationship with the metal exposure and CKD. So now we are going to start to, if someone had any question for Anna, you can unmute your microphone and ask Anna. También recuerden, la pueden hacer en español. Ana habla español y podemos traducirla igual.
Well, Anna, thank you. Thank you for your presentation. It was really, really enlightening. Well, we are working in Panama somehow in the same region, in the same topic. I have a question in my mind right now. We are having some issues with the change in the climate patterns, you know. So rain is affecting the quality of underground waters, and we are having some kind of switch between summer and winter. So how far do you think could be the effect of combining other metals like nickel, iron, and manganese, let's say, in this accumulative effect that you are showing off?
I think it's very important. I only highlighted the findings for the metals for which we have the best effects. But I think it's going to be very important to do this metal mixtures analysis. I said we need to be careful because the interpretation of the biomarkers can be difficult, but I think if you can combine this environmental sample data information with biomarker data, I think it's going to be very important to do. And the good news is with the new tools in biostatistics that have been developed also in the last few years, this is becoming much more easier. So thank you for that point. I think it definitively is very important to study. And what you mentioned about the impact of the changes in climate to exposures, those changes tend to be slow. That's what people say. But if you're changing very rapidly the groundwater, maybe it's getting much faster. Is the way, like, you know, people are being surprised now with how fast these temperature heat waves are happening. So I think maybe we are seeing these changes at a much faster pace than we even anticipated. So looking at the impacts for metals in groundwater and how populations are exposed to this, I think is very important, and we need to really pay attention to that. So thank you. Yeah.
Thanks to you, Anna. Thanks.
I see that there are lots of comments, but I don't know if it's relevant if there are any questions, maybe not.
No, there is no any question on the
Okay.
chat yet. So, and
People are
monitoring the
saying
chat.
hello, where they are coming from.
Yeah.
Very good.
Exactly. Is there any other question for Anna?
Hi, Anna. This is Andres. I actually, kind of what you mentioned about the ability to measure multiple metals now in blood, but having that statement that maybe that's not the ideal matrix for all the single metals. So in the ideal case scenario, what do you think would be best moving forward? Like, for example, for the CURE consortium, right? What is it the water to look at chronic exposure? Or do you think the biomarkers might be sufficient to be able to test that hypothesis?
I think definitely the biomarkers are a great place to start, and it's easy, relatively easy to collect. And I mean, within a cohort study, it's relatively easy in the sense that you are already collecting blood and urine. So that's great. So I think it's important to include them. I think my concern, maybe for the toxic metals, it's easier, but again, lead in urine, we don't know what it means. So if you have a panel, it's easy to measure lead in urine, but do you just include that in your BKMR and then hope for the best? I don't know. So I think maybe adding some tagging, I think we need to be tagging kind of information to these metrics so that we can be able to say, okay, good, these are the priority metals for the urine, these are the priority metals for the blood. And maybe it's still too early for us to do that, in the sense that we don't have, now we see often inconsistent findings across cohorts. But I think it's because we have small sample sizes, but as we pull data, I hope that we can start seeing more clear signals that tell us which markers are better in where. But I think we can do this. It's like when you do an untargeted analysis, I think it's okay to do it. But what I think it should be, we should not be as naive as to report it without adding some caution and trying to think carefully. For instance, if you study zinc biomarkers are very complicated, and they're not telling you anything about zinc intake. So so sometimes I see these things published, and then I'm like, oh my goodness, these people have no clue what they are doing. And so I think that's where we have to be very careful with interpreting the biomarkers. And but it's exciting. I feel this is an incredible time because we have the technical capability, and then we are adding the content knowledge. I think the knowledge that we are generating also is growing really fast. So I hope that this is going to help us to really have real knowledge advancement. So that's exciting. But thank you, Andres. Yeah.
No, thank you. That was a wonderful lecture. Thanks.
And you know that we measure metals in our cohort. We did it. We compared the metal level during the baseline at the second study visit in our cohort. Right now, we are planning to replicate the analysis in the entire cohort. What would be your recommendation for this? We had more than. We had a big biobank because we have been conducting this cohort study over 7 years now. And we had 1,000 participants in this cohort. So we identified the incidence of CKDU in this population is 1%. Right now, and also we had identified people with rapid declining GFR. So we want to again to measure the metals because we want to know if there is any relationship with the metal levels and the decline of GFR over time.
That's great. Marvin, I was looking, I did a search on your name, metals and kidney, to see if I found, is your study published already?
Yes, it was published in 2019.
Okay.
It was the first one.
Okay, very good. So
in that paper, we measure as well pesticide, 12 nephrotoxic pesticide, and they are metabolized. And we haven't we didn't find any relationship with the rapid decline. in the
Good.
comparing the first and the second study visit.
I am going to look. I look at your paper and see if I have any suggestion. But I think if you can do some repeated measures of the metals, that will be great. And then I think to look prospectively, adjusting carefully by baseline EGFR, I think it's also important. So I recommend really to always do the prospective analysis, adjusting for the baseline EGFR, because that should help quite a bit. Otherwise, it's always very tricky because of the reverse causation that I mentioned earlier.
Yeah, I totally agree with you. The reverse causation is a big issue in these studies.
Yeah. So that's why I think I forgot to mention that when answering to Andres earlier, the environmental samples have a role, because if have multiple roles, the environmental samples. One is to identify the source. Where is it primarily coming from? And that can be helpful for mitigation and prevention. But the other reason why it can be helpful is because they are not affected by reverse causation. So I think the combination of environmental and biomonitoring, I think should be helpful.
Also, we try to adjust it by seasonality here in Nicaragua. You know, we had 2 seasons, the winter and the dry season. So probably the metal could be changed over the season, because mostly regarding pesticide, no metals, but pesticide. Yes, and we didn't find any highest level between these 2 seasons.
Mm-hmm. Very good.
So I think I don't see any more question in the chat, and I don't know if somebody else want to do any questions.
I am available also if people have questions in the future or want to just ask a question for some of the research they are doing. I am happy to contact. And
yeah, just a final question, Anna. Are you thinking about an open collaboration with Latin America, let's say Panama, Nicaragua?
I mean, as part of the CURE consortium, I am involved because I am involved through the India field side, but obviously I am involved for the whole consortium, and I am very excited to be part of that. And then I am very happy to help with other things that you might be going on. If you have questions or something, anything, as much as possible, I like to help.
I appreciate that. Thank you.
And the SALTRA group, the UNA SALTRA group is collaborating in the CURE consortium as well. In Panama, Professor Ildaura Patiño, Acosta de Patiño is working in the CURE proposal as well. And so, as Anna said, we are planning to do a big study in Central America and India. So we will be collecting environmental, biological samples for testing all the hypotheses that have been proposed related to CKDU. So in the future, we will have more evidence about the metals role on the pathway of CKDU.
Mm-hmm.
Well, and now it is 8 p.m. So we are ending this seminar. Thank you so much, Anna, and thank you to everyone that joined this seminar. And it was a fantastic lecture, and I think we had learned a lot about the role of metal roles on the CKDU. And
thank you. Okay.